Welcome everyone to the seminar in socio-epidemic mathematical modeling. Today is the Pi Day, and we have a great speaker to celebrate the Pi Day. So earlier today, I did this naive thing of going to Professor Maini webpage and try to find some information to introduce you, Philip. But this was very inspiring, but I realized I cannot fairly summarize your achievements. So I will just say Professor Philip Maini is a professor at the University of Oxford, a great mathematical biologist, and is a great honor to have you speaking at to the Italian community with a talk on modeling collective cell movement. So I will leave you the floor. Thank you very much again.
Yeah, well, thank you, Francesca, and thank you very much for the kind invitation. And I should say that if at some point you can't hear me or something, then please do let me know. So I'll be talking about modeling collective cell movement, and I'll be talking in the context of something called the cranial neural crest cell migration. And first of all, it's important to say who the collaborators are. So I've been working with Paul Klesse, who's an experimentalist, and he's been at the Sowers Institute for Medical Research for a very long time. He's now recently moved to Children's Mercy in Kansas City, and his colleague Rebecca McLennan, who worked when they were at the Sowers. And the main collaboration is with Ruth Baker, who's my colleague here in mathematics, and a number of the people who've done the bulk of the work, a number of graduate students, Louise Dyson, who's now at Warwick actually working on epidemiology, Lina Schumacher, who's now at Edinburgh, Raza Ginuati, who's at Vilnius, and Duncan Martinson, who's a postdoc here. So what is the neural crest? So one thing that a lot of people don't realize is that many of our cells begin in certain parts of the body, and they have to actively move to where they're supposed to be. So, and the neural crest is a great example of this, because these are different regions, roughly speaking, along our backbone that very early in development, these cells have to move to different places. So, and they form all sorts of different structures, muscle cells, pigment cells, etc., etc. And we have 4 different neural crests. And here they are: cranial, which is one I'm talking about, which is skull; trunk, which I mentioned briefly, which is obviously the going to the digestive system, the nervous system, and the cardiac system. So here is a cartoon. This is the head. Or maybe I should go into use one of these laser pointers. Hopefully you can see. This is the head. This is the tail. And these cells undergo something called epithelial to mesenchymal transition, which is EMT, which is something that cancer cells also do. So epithelial cells are cells that are stationary, that take part in forming sheets. Mesenchymal cells are cells that move. So these cells, who originally epithelial, become mesenchymal and then actively migrate. And the ones we're looking at are the ones that are going to be these ones, branchial arch 4. Oh, sorry, I've got the wrong thing. These ones here, they go to branchial arch 2, rhombomere 4 to branchial arch 2. And of course, it's 3-dimensional, so they're going like this. And the domain is growing at the same time. Why should we study this? Neural crest in particular, that's important for proper development of the face and neck. And altogether, there are 66 related diseases to neural crest. So 1/3 of birth defects arise from some issue with neural crest. And the neural crest are very similar in behavior to highly aggressive cancers. So it serves as a really powerful paradigm, and it is experimentally tractable. And there's a summary of this in this paper that was published in 2020. And what I should mention is that invasive cell motion has been widely studied in the context of developmental biology, but also in, for example, wound healing, which is repair, or disease like cancer, or animal movement. And in fact, recently, from last year, from July until December, we ran a program called the Mathematics of Movement at the Newton Institute in Cambridge, where we brought together people who model how animals move and people who model how cells move and the collective behaviors to try and see what are the common processes and what are the differences. So I'll first of all talk about the gut and give a review. So in the gut, there's something called Hirschsprung's disease. And this arises when, so the gut is growing and the neural crest cells are moving along to cover the gut, to coat the gut. If they don't reach the end of the gut, then the mechanics of the gut end up being abnormal. And this results in huge development of the gut. And this very nice research paper from Kerry Landband and Matt Simpson, who are both mathematicians, and Don Newgreen, who's a biologist, gives a lovely review of this. And it's natural to assume that if your cells are not reaching the end of the domain, that they're not moving properly. And so it's natural to see, are there any genetic abnormalities to do with movement? But what they did was to write down a model system which accounts for the fact that you've got proliferation as well. So here they look at experiments where you've got a donor, which is N1, and you've got host, which is N2, the cells, and then you've got diffusion. Consider the domain as being one-dimensional. You've got logistic growth, and you've got advection due to the growth with the velocity v, which is the velocity at which the domain grows. And as you can see from this equation, if we didn't have N2 and we didn't have the growth, essentially this is Fisher's equation. And we know that for Fisher's equation for traveling waves, not only is the diffusion important, but the proliferation is equally important. And they did an analysis of this model, and what they found was a parameter space in which you would get full colonization, which means you fill up the gut, versus areas where you got a small colonization, so Hirschsprung's disease. And what you can see here is that not only does it depend on cell motility, it also depends on the number of cells. And the number of cells, so it's important then, this extra biological insight from this model is to do with the fact that the cell number is also important. And then another model that's been opposed is called contact inhibition co-attraction. And the idea of this, and this is work from Roberto Mayor and his group, and this paper is with Karen Page, who's a mathematician. The idea here that they have is that cells, one type of cell attracts another cell type, but that cell type inhibits the movement of the other cell type. And so what you end up getting, one cell type gets attracted to the other one, and that moves the other one away because the other one is inhibited from that cell. And then that is repelled by that cell. And in that way, you get movement. And here's a model that they did, which is quite a detailed mechanical model, where they have cells. They model the shape of the cell, preferred direction, etc., etc. And what they show here is that they've got 4 cases. So you've got contact inhibition of locomotion, which means that when cells contact each other, there's an inhibition, and then you've got co-attraction. And what they're trying to model is how do a clump of cells move? And what you find here is that you need both these processes to get this clump to move. And this work has been mathematically analyzed with Kevin Painter, who now actually is in Torino, used to be at Heriot Watt in Scotland. And here they look at a diffusion, and then they have a long range model to account for this co-attraction and inhibition, where this is a kernel that describes, this is the how the long range effects impact the movement of U. And you can see here, they find a rich behavior in this non-local model. So if we go back to our model, these models that I talked about are not applicable to the cranial neural crest for 2 main reasons. The first reason is there's very little proliferation, which means that the movement is not driven by proliferation in cell number. The second thing is it's not a clump of cells that move. Instead, it's a chain of cells that move. So we have to come up with our own model. So we're going to simplify things. There's a curvature here. We're going to assume that doesn't matter. There's also a depth, which is about 3 cell widths, and we're going to assume that's negligible. So we're going to consider this as being a rectangle that is constant width in the y direction, and it's growing in the x direction. And cells are coming in at one end, and they have to get to the other end. Here's a chick. Here's the head. Here's the tail. Here's rhombomere 4, and the cells you can see are moving towards branchial arch 2, and this distance is about 1 millimeter. And here's a close-up of these cells. So here's some experimental data. Over the course of about a day, the pathway grows from about 300 microns to about 1300 microns. So it's about 1 millimeter. So what we do is we fit a curve to this, roughly speaking, logistic type curve, and that's our growth function of the for the domain. And so clearly, if the cells were just diffusing, well, A, they would not be able to reach that distance in one day, but more importantly, they would not go in a coherent way. They would not be a chain. What our biology colleagues had observed was that the tissue produces this growth factor, which actually is very common in cancer dynamics. It's called VEGF. And the cells consume this VEGF and they create a gradient. And then they move up the gradient, so it's a chemoattractant. So the hypothesis was that this leads to a bias random walk, and that will do two things. It will speed up the movement, and it will cause the cells to move in a coherent way. So those were the hypotheses we were going to check. So we wrote down a reaction diffusion PDE model for VEGF with a saturating source production, and we modeled the cells as individual discs with a center that are sinks for the VEGF. So we go a bit further here. Here's the VEGF C. We rescale so that the diffusion coefficient in the X direction, we rescale it so it becomes 1 over L squared, where L is this logistic growth term. This is dilution due to the growth. This is the production. And this is consumption, where basically we say it's a Gaussian with a maximum at the center of the cell. So this is just a phenomenological consumption term that we put in the model. We need to have boundary conditions. And when we started this project, we had no justification to decide what the boundary conditions could be. We started off by putting homogeneous Dirichlet boundary conditions on the chemoattractant. That meant that the gradient was inward pointing, so no cells left the boundary. Later on, we used zero flux Neumann conditions so that cells don't leave the boundary. The key results that we got don't matter, don't depend on these two conditions. But I'll come back to these conditions a bit later. We consider the cells as discrete entities that are volume filling, and they stochastically sense the gradient. And if they find that nearby, in the direction they sense, the chemical concentration is larger than the chemical concentration at the center, taking noise into account, they then move in that direction with a fixed speed. So it's the simplest possible way to model this. And the reason why we don't use a PDE for the cells is because there's a very small number of cells. So stochastic effects are important. And what we find is that it doesn't work. So here's the domain, the beginning, 300 microns, and starts to grow. Cells come in. These lines are where they're stochastically sensing the density and moving up the gradient. And what you see here is at the start, it's actually quite good movement. But if you also notice here, the VEGF is being depleted, which is something they observe experimentally as well. So the VEGF disappears and these cells get left behind. So it doesn't work. If we take this sort of analogous model in a PDE context, we get the Keller-Siegel model, where N are cell density. This is movement up the chemoattractant gradient. C is the chemoattractant diffusing, and then has got kinetics linked to the consumption from the N. And this model, it was shown, and it's very easy to show, will only will exhibit quite easily a pulse of cells moving, but for it to give a train, like a Fisher wave, that has to be singular. In other words, the chemotactic sensitivity has to increase as 1/c. And that is not biologically realistic, because what that is saying is that as the signal goes to 0, you have better, the signal is better. And that obviously doesn't happen biologically. So the student working on this, Louise Dyson, she thought the following. She said, well, if these cells at the back here that are stuck, maybe they could follow the cells at the front. So instead of looking for a chemotactic gradient, if they sense the cell at the front, then they stick to it and they move with it. So the idea is that you have two cell types, cells at the front that are leaders, cells at the back who are followers, and then you do get these chain-like migration. So we asked our biological collaborators, are all these cells the same? And they said they would do some genetic analysis to answer our question. Okay, so that just summarizes what I just said. And what they found when they did their analysis was that there were significant differences in gene expression between the cells at the front and the cells at the back. And it was more complicated than that. So they split it up into 4 regions. They looked at key genes of interest, and you can see, I'm not going to go through all the detail of the genes. There are 96 genes. You can see there's very different expression patterns. And crucially, what they found was that the cells at the front upregulated VEGF receptors. So they really, and they upregulated genes that resulted in the formation of enzymes that broke down the material they were moving through to enable them to move better and to move up gradient. And the cells at the back upregulated cadherins, and those are stickiness, adhesive. So the cells at the back were much more adhesive than the cells at the front. So it was consistent with their model hypothesis. So then what we did was a number of validation. You can see this collaboration's gone back like as 12 years now. We've been collaborating. And these validations consisted of them doing experiments where they would basically do cut and paste experiments, cut certain parts from a host and replace them with certain parts from the donor. And we did the simulations that they could do experimentally. And during that time when we were doing this, we didn't communicate with each other to make sure that we didn't give away any secrets and to make sure that these were independent tests. And then we would meet up, they would show the experimental results, and we would show the model predictions. Now, many of the predictions of the model, the mathematical model, agreed with the predictions in the chick, but I feel that sometimes you learn more when the experimental model and the theoretical model don't agree. And this is one example. What happens when you take the followers from a donor and you stick them to the front of the host? What our model predicts is that when you stick these cells that are followers, you put them well ahead in the front, like here, these red cells, then they have no cue because there's nothing to stick to, so they move randomly. Here's the experiment. So here's the host in blue, and here's the donor in red. And what you can see happening here is the donor is actively moving. This is not random movement. This is bias, random walk. So in other words, the donor cells, the followers, have become leaders. They've switched phenotype. And they respond to the VEGF, they consume the VEGF, and they move up a gradient of VEGF. So the next experiment, natural one to do, and you do a mathematical model to figure out what the result would be, although we did do the mathematical model anyway. What happens if you now put VEGF along the route that the cells take? Well, common sense tells you these cells are too far away, they'll continue moving. These follower cells will become leaders now due to this environmental effect. They'll switch their phenotype and they'll move towards the VEGF. And that's what happened. You get a breakage. And here's the model showing that breakage as well. So another question our collaborator asked was, how many leaders are necessary? And this is the work of Linus Schumacher. So we said that because the parameters we chose in our model are sort of, this has not been done by model fitting, et cetera, so the parameters are gross approximations. So we can't make such a quantitative prediction, but we could make qualitative predictions. So we asked the question, what would happen if we increased the number of leaders? So here's normal, and now this is now with zero flux boundary conditions. The leaders are in purple, the followers are in this sort of orange color. And now you see, and then we add up, integrate across, and then we plot. And here is normal, so to speak. And you can see we get invasion. And then we redo the experiment where we increase the number of leaders. And you see we still get invasion, but with fewer cells. So the model prediction is, if you increase the number of leaders, you will get invasion, but fewer number of cells. And here's the experiment. Here's normal. And then here's the application of some chemical that upregulates genes that are known to lead to the leader phenotype. And what you see, you get invasion, but fewer number of cells. So qualitatively, it agrees with the theoretical model. Quantitatively, you can see it doesn't agree. But we didn't expect it to agree quantitatively. And now we come to boundary conditions. And this is an old slide because this one's already been published now. It's published last year. Let's now look at this corridor in more detail. So we put zero flux boundary conditions along here, and we had no physical justification for doing so. When you look at the images, there is no physical barrier. But it is known that cells do not move into these regions. So we asked the question, okay, here we've got total zero flux boundary conditions. Here we've got no boundary condition. We have the boundary conditions. We put them at the end. We think of these as being internal boundary conditions. And here we have a partial boundary condition. So then we ask the question, if we vary the length along which we have this internal boundary condition, how does that affect the behavior of the cells? And we characterize this by saying that if all the cells keep in this region, then that's very good. If they deviate a bit, then that is okay. If they deviate a lot, then that is really bad. So then Raza did these simulations. These two you would expect. The results are what you would expect. Here, with the zero flux boundary conditions, you get perfect invasion. Here, where there's no internal boundary condition, it's a complete mess. Here's an example here where it's actually not too bad. And so because there isn't enough time, I didn't go into other types of following that we looked at. We looked at tunneling because we found out that the cells at the front also secrete a chemical that allows them to make holes in the extracellular matrix and a tunnel, and so the followers can move through the tunnel. But let's look at this as the model that we've been talking about. Let's look at this one. So here we have length of internal confinement boundary. And here we have the fraction of cells in the different regions. And what we can see here is that this fraction, about 3/4 of the cells, are in the right part of the domain, but these are in the wrong. But by the time you get up to, roughly speaking, 500 microns, virtually all the cells are in the right part of the domain. So what this model is predicting is that you need to have some chemical that is next to the entrance as the cells come into the domain, but only needs to be expressed about halfway, up to halfway along the domain. And then this is the work here, it was published a few months back. They found such chemicals, and they found them expressed not fully along the corridor, but focused near the entrance. And they're called collect 12 and trail. And here you see an experiment where you put streaks of collect 12 along here. And then you start off with the cells along here, and you see after a while the cells have moved away from the collect 12. So collect 12 is an inhibitor. And what trail does, trail slows cells down. So if cells are heading in the direction of trail, which is the wrong direction, they slow down. And presently, we're about to publish a paper, about to submit a paper actually, where we show that having these two processes effectively gives you a zero flux boundary condition. So one thing that I didn't point out was that in our model, going back now to the model where don't worry about partial boundary condition, be full boundary condition. And remember, this one didn't work. The leader-follower did work, but look at here. These leaders have got too far away. So they're no good. They've broken away from the chain. So the question that we asked was, can the velocity of these cells be controlled so that they don't break away from the rest of the chain? And so Paul and Rebecca and the collaborators went and they looked near the origin, and what they found was the expression of something called DAN. DAN is a protein. And here you can see it, this red is DAN, and it's expressed right next to the cells coming out from the neural tube, delaminating from the neural tube. And here we have an experiment. Here's Dan, 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 and no Dan. And what you can see here is that the cells are two things that happen. One thing is they are repelled by Dan, but another thing is they move more slowly when they're on Dan. So that would mean that the cells, the leaders that are coming out would be slowed down. But it also means the followers would be slowed down. But if we look here, as this process continues, it appears that the expression of Dan is no longer visible. So could it be that as these cells move through Dan, they degrade Dan? So that means when the followers come in, the followers don't have any barrier. So the leaders are slowed down, the followers are not. And remember, I told you that neural crest cells share a lot of properties with some of the most invasive cancers, such as melanoma, for example. If you confront melanoma cells with Dan, they are repelled by Dan. So in the long run, looking way down the line, this could be some way in which you might be able to control the invasion of tumor cells of certain types. But we haven't explored that further at the moment. So there's a question of how do followers follow? We've done stick to the cell in front, the chain migration. We've also done that it could follow a path cleared in the tissue by the cells in front. That's called tunneling. And that tunneling is in this paper here, where Paul and his experimental colleagues showed that the cells at the front secrete aquaporin 1. What this does is it causes the filopodia, so the finger-like protrusions that the cell pushes out, it makes those quite strong, and it can push a hole through the tissue it's moving in. We could also have that it's a chemical signal. Cells at the front could secrete a chemical that causes the cell next to the front to move, and then triggers them to produce a chemical that causes the next ones to move. And in fact, that's something that we're actually looking at the moment, modeling that at the moment in the context of a PDE system. But then there are other signaling cues. And this is work of Duncan Martinson. For example, there's something called fibronectin, which is an adhesive molecule. And at the beginning, these are expressed as lumps called puncta. And as the neural crest cells move over them, they remodel them into strips. And it's known that the followers, using something called contact guidance, are then guided to move in the same direction as these strips. So for this model, we decided, let's just focus on that aspect. So let's not worry about the growing domain. Let's not worry about chemotaxis. Let's start off with leading cells here in black. They come out, and these little spots here are puncta. And these black cells, as they move over the puncta, they create an alignment signaling cue, and the cells at the back stick to them. So in this mathematical model, each cell has a sort of domain in which it sort of sums up the different cues. So there'll be the directional cues, which will be a force pushing it in the direction of the fibronectin puncta. There'll be cues due to the fact that there are other cells and they're pushing them because of volume. Then we take the net force and we move the cell in that direction. Okay, so unlike the previous model where we just say there's a rule over how the cell moves, here we integrate the signaling, get the resultant net force, and move the cell in the direction of that force. And what you can see here, as we go from 180 to 360, 540 to 720, we get really good chain migration. This is a stochastic model. So now we rerun again, and we get this type of behavior. They go off in the wrong direction. So remember, here there's no chemotaxis, so these cells do not have a directional cue to get them to move in the right direction, nor do we have the boundary cues. So one thing that we're going to do in the future is we're going to put those cues in, and we're also going to grow the domain, because we also feel that as the domain grows, that could be a cue. So Duncan did a huge amount of analysis here by taking into account the different forces that are happening here, and because we don't know what the contribution of each force is, he varied the contributions in order to see which was most important. And using global sensitivity, what he found was that if you upregulate contact guidance, that's good in all aspects. Haptotaxis, which is a process where you move up an adhesive gradient, and fibronectin is an adhesive gradient. If you focus on that, cells cluster together, which means they do not move as a coherent stream. If you make volume exclusion very important, this as something that we didn't expect, it has a bimodal effect. Because we might think that volume exclusion, which means that cells push each other, would enhance migration. But in fact, we found, well, it can enhance migration under certain conditions, but it can hinder migration under other conditions. And we hadn't expected that result. And then he concluded that contact guidance and matrix remodeling are the best ways. So in other words, if you have the cells moving over the fibronectin and they remodel the matrix and the following cells are directed by that, you can get collective migration. So what we've seen here is that this rather seemingly simple problem of how do you get a few hundred cells to move 1 millimeter in one day is actually really very, very complicated. You have to have phenotypic heterogeneity, which means you have to have different cell types. You have to have control of the velocity of some of the cells. You have to have signaling from one cell type to another. And we've looked here at signaling in terms of sticking, in terms of creating a tunnel, in terms of creating a sort of aligned matrix. And probably what is happening is all three of those things. And probably even more things are happening. And then we've also seen that to keep the cells in the corridor, we need certain chemicals, and those chemicals need to behave in a certain way. So there's a natural question to ask, which is, we've ignored curvature. Does curvature make a difference? And we're presently writing up a paper which shows in a simple example of this PDE model, the curvature that's needed to make a difference is much more curved than the curvature of the embryo for the neural crest. So we sort of feel that we've justified that simplification. The simplification of going from 3D to 2D, we haven't justified that. That would be an interesting thing to look at. Cells could swap neighbors and move up or down. We haven't got around to doing that. But there's other things we haven't taken into account. We have assumed that the growth, domain growth, is spatially uniform. We just have it changing in time, not in space. We checked that experimentally, and that's not true. It's not spatially uniform. In this model, we did a few sort of test experiments on the mathematical model of what would happen if we had growth being non-uniform in some different places. How does it affect the behavior of the system? But we haven't done that in the detail that it needs to be done. What makes it even worse, more complicated, is that the cells induce the growth. And that's something that it's not clear how they induce the growth or when they induce the growth. So one thing we're hoping to explore is, by taking different hypotheses of this, can we find out what is the strategy that these cells are using when they're growing the domain? And I talked about there being two phenotypes. There are not two phenotypes. There's a gradient of phenotypes. And that work is ongoing. And that then becomes really very, very complicated, because now you've got cells that are moving in physical space and their behavior is changing in phenotypic space depending on the signals that they're getting from the domain. And one thing I'd like to point out is that there are many similarities between this and cancer and angiogenesis, because in these two things, you also have leaders and followers, and you also have signals going from leader to follower. You also have VEGF. You also have cells switching phenotype. This is also relevant in wound healing and also relevant in animal behavior, where you have leaders and followers, where you have signals from informed individuals, the leaders, they transmit signals to the followers, so the followers follow, and you have the switching of leaders and followers. So what we see here is that in the context of collective movement, there are a few processes that seem to be fundamental. And then other processes depend on the particular problem that you're looking at. And just to finish, what I find amazing is that we can send people to the moon, which is many hundreds of thousands of miles away. We're talking about sending people to Mars, yet we don't understand how the cells in our body move a distance of 1 millimeter. And that to me shows the astonishing complexity of biology. It also illustrates the huge amount of opportunities there are for modelers in this context. So I'll end there and thank you for your attention.
Thank you very